Hi students, how are you all? Hope so you're all very much fine. So previously we have studied about pollination and fertilization in the session 2 class. So this is our biology session 3. In this we are going to deal with the chapter 6. So the chapter 6 deals with the seed, structure and germination. So previously we have studied about pollination and fertilization, in which the fertilization process occurs between the pollen grain and the ovule of the flower, and it forms the embryo. So after the formation of embryo, what's going to happen? So this is a connectivity to the next lesson, structure and germination of seed. Now let's get into the So in this lesson we are going to cover the topic what is seed, the characteristics of seed, classification of seed, structure of seed, the bean seed and maize grain, germination, conditions and experiments on germination, types of germination. So we should know the definition for fruit, seed and grain. If you take fruit, we very well know after fertilization some of the changes going to take place for the floral parts of the flower. So the ovary changes to fruit, the ovule changes to seed, the ovarian wall changes to fruit wall. So here what is fruit? A fruit is an enlarged ripened ovary which is having a fruit wall enclosing the seed, and this fruit wall is going to protect the seed and also helps in seed dispersal. Seed, it is a ripened ovule which is going to contain the embryo. This embryo is going to grow into a new plant. Grain, so grain, it is a combination of fruit wall and seed coat. The fruit wall and the seed coat are fused together, which is going to act as a protective layer. So it is called a grain. So this is what the definition for fruit, seed and grain. Characteristics of seed. So first thing, the matured ovule, the fertilized ovule is a seed, and it is a tiny living plant called embryo. It's a formation of sperm nucleus plus egg nucleus. So when the sperm nucleus and the egg nucleus fuses together, it forms an embryo. From there a tiny living plant is going to come out, and the embryo, after the formation of embryo, it's going to remain inactive, that is dormant stage, inactive until it is exposed to the favorable condition for the growth. And seed also contains a food material which is going to give the nourishment of the embryo during the germination process. And this embryo can withstand unfavorable conditions like temperature, drought, etc. more than 100 of years. Some seeds will live more than 100 of years, some seeds will live less than 100 of years. Okay, so these are some of the characteristics of seed. So seed, it is a fertilized ovule and it is a tiny living plant and contains the embryo, the formation of a sperm nucleus plus egg nucleus, and it remains inactive until it's going to expose to the favorable condition, and it also acts as a food material for the nourishment during the germination process, and it can withstand the unfavorable conditions. Next is classification of seeds. So based on the presence of cotyledons, the seeds are classified into two groups. So cotyledons means seed leaf, we very well know that. So based on the presence of seed leaf, the seeds can be classified as monocotyledon and dicotyledons. Monocotyledons, one seed leaf, that is the seed which contains one seed leaf is called monocotyledon. In short, it is called as monocot. Example, maize. And the seed which is going to contain two seed leaves is called dicotyledons. Example, bean. So based on the size, the seeds are classified as large, small, quite large. Large seeds, example, you can give coconut, and quite large is the seeds which is present in the watermelon, mango, etc. and the small seeds, the poppy seeds and orchid seeds. And the next is based on the presence of endosperm, the seeds are classified as albuminous and ex-albuminous. Albuminous is also called as endospermic seed, and ex-albuminous is called as non-endospermic seeds. In the albuminous, the cotyledons are very thin and membranous, and in ex-albuminous, the cotyledon is thick and fleshy. So we have dicot albuminous, monocot albuminous, And as usual, the X albuminous also can be classified as dicot X albuminous and monocot X albuminous. The examples for dicot albuminous seed is poppy seed, monocot albuminous cereals, dicot X albuminous pea, and monocot X albuminous vallisneria. Okay, so I have given only one example for each. So you can go through your book for some other example. So this is what the classification of seed. And the next is the bean seed. So we are, I said in the topic covered slide, that is the structure of the seed also we are going to see. So in that first we are going to see the structure of bean seed. So there are different types of bean seeds. Most probably the size of the bean seed will be kidney shape, both convex and concave. And the next is seed coat. So the seed coat can be differentiated in two layers, testa and tegmen. Testa, it is the outermost hard brownish layer, and it is going to protect the seed from the physical and mechanical injuries and mechanical barriers. Tegmen, it is the protective layer which is present very next to the testa. Okay, so seed coat, it consists of testa and tegmen. Testa is the outermost layer and tegmen which is present very next to the testa, and it is a inner layer. Tegmen is the inner layer. Okay, so this is what is seed coat. Next is hilum. It is a whitish oval scar which is present in the concave side of the bean seed. If you take the bean seed, in middle you can see a small concave oval in the bean seed, that is called a scar. Okay, so it represents the spot where the ovule was attached to the ovary wall through the placenta. So when we are studying about the parts of the flower, in that we have studied placenta. What is placenta? Where the ovule is going to attach to the wall of the ovary. So where that ovule is attached, that point is called the hilum, which is a whitish oval scar. Next is micropyle. It is a tiny pore which is present next to the hilum. It's a small opening present in the seed which is going to helpful in absorbing the water and also diffusion of gases for the respiration. So micropyle, and it is also useful in the entry of pollen tube during the fertilization process, through which the sperm nucleus is going to enter into the egg cell. So this is what the structure of bean seed. So can you able to see the picture here? I have clearly given here hilum, micropyle and seed coat. Here on separating the two cotyledons. So bean is a example for dicot seed. So when you are going to separate the two cotyledon, embryo is seen over there. So this embryo contains two parts, that is radicle and plumule. Radicle on further development, it changes as root. Plumule on further development to shoot. So radicle develops to root and plumule develops to shoot. So the picture is given clearly for you, your children. Hilum, the small whitish scar over here, and next to the down to the hilum, a small point, a micropyle is present, and seed coat is marked here. And here epicotyl, hypocotyl, radicle, that is the brownish layer is there, no, that is the root which is going to develop, and seed coat, cotyledon is marked. So this is what the structure of bean seed. Next is example for monocot, maize grain. So in the introduction part I have said grain is a combination, that is fusion of fruit wall plus seed coat. Okay, so fusion of fruit wall plus seed coat, it forms a grain. A light colored oval area is the embryo present in the maize grain. So if you take a maize grain, you can able to see a light colored oval area. So that is the embryo, where that is the place where the embryo is present. The remaining portion of that seed is called endosperm, and this endosperm and embryonic layer, okay, the embryo and endosperm layer is separated by a thin epithelial part. Okay, so the small oval region is called the embryo, and the remaining part is the endosperm. This endosperm and embryonic layer are separated by a thin layer called epithelial part. The endosperm is rich in protein. The endosperm is rich in protein. It is called the alluron layer. And embryo, it is a single cotyledon called scutellum, where the radicle and plumule is going to present. Okay, so endosperm, it is rich in protein, and that rich protein is called alluring layer. It is the outermost layer which is rich in protein, and it is called the alluring layer. And the embryo, it is a single cotyledon. It consists of single cotyledon which is called scutellum, consists of radicle and plumule. So here in maize, the radicle which is grow towards a pointed end, it's called a coleorhiza. The radicle is towards a pointed end and is enclosed by a protective sheath. Okay, so radicle which grows towards a pointed end, it is covered by a protective sheath called coleorhiza, and the same plumule towards the upper border, it is covered by a protective sheath called coleoptile. Okay, so this is what the structure of maize grain. So the next process we are going to see is germination. What is germination? The process of formation of a seedling developed from the embryo. So this is what the germination. So as I said before, the seed is going to remain dormant, that is inactive, until it's going to exposed to the favorable conditions. So though it's going to remain inactive, the small chemical reactions, chemical activities are going to take place, and the diffusion of gas is also going to take place within the seed itself. So once it's going to reach the favorable condition, it starts germinating. So what are the conditions required for the germination? So there are three main conditions required. One is water, the next is suitable temperature, and the next is oxygen. First, water. So we very well know the seed and the plant are going to get the water from the surroundings, the environmental surroundings, from soil and from the natural conditions. So the microbial which is present in the seed is going to helpful for the embryo to absorb the water. Once the microbial is going to absorb the water, what happens? The seed swells. The seed swell and the seed coat rupture, allowing the elongating radicle to come out and form the root system. So once the water is absorbed by the seed swells and the seed coat will rupture, the radicle will come out and the radicle will develop into a root system. And this water is also necessary for some chemical reactions. And the food stored in the cotyledons or endosperm are converted to diffusible form, dissolved and utilized by growing embryo because of with the help of this water only. And the next is suitable temperature. So the seed cannot withstand both low and high temperature. So both low and high temperature are unsuitable. So moderate temperature is must. 25 to 35 degrees Celsius is a moderate temperature for a seed to germinate. And the next is oxygen. So oxygen is needed for cell division and cell growth. In short, we can say the respiration process. Oxygen is needed for the respiration process. Optimum temperature is needed, and water also required for the germination process. So these are the three major conditions required for the germination process. The next is types of germination. So before entering into the types of germination, we must know what is epicotyl and hypocotyl. Epicotyl. The epicotyl means the region of the axis between the point of attachment of cotyledon and the plumule. Okay, so the region of the axis between the point of attachment of cotyledon and the plumule, it is the epicotyl. And hypocotyl, it is the axis below the cotyledon. So epicotyl, it is the attachment, point of attachment of cotyledon and the plumule. It is present the axis between the cotyledon and the plumule. Hypocotyl, it is axis below the cotyledon. So epicotyl, hypocotyl. So based on this, the germination has been classified as hypogeal germination and epigeal germination. So this epicotyl elongates and it grows underground without coming out. When the epicotyl is going to elongate and grows underground, that type of germination is called hypogeal germination. And the hypocotyl elongates above the ground, it is called the Apigeal germination. So in short, how you can say cotyledons remains underground is a hypogeal germination. Cotyledons pushed above the ground is called the apigeal germination. So here I have given in the picture the hypogeal germination and apigeal germination. See here, in hypogeal germination the cotyledons are remain underground, and the apigeal, the cotyledons are growing above the ground. So this is what the diagrammatic representation of hypogeal germination and apigeal germination. So the next type is viviparous germination. So what is viviparous? Producing live young ones. Just like mammals, how it's going to produce the young ones directly, give birth directly to the young ones. Likewise, some plants are going to give birth, that is germinate the seed by live young ones. Okay, so here the seeds germinate inside the fruit while it is still attached to the parent plant, and after it falls from the parent plant and grow into a new plant, and hence it is called a viviparous germination. The example for viviparous germination, you can take a rhizopora. Okay, and the next is experiments on germination. So here 4 experiments given for germination process. That is, the experiment to prove that water is necessary, suitable temperature is necessary, oxygen is important, and 3 bean seed experiment. So all the 4 experiments are very much clearly explained in your book, page number 50 and 51, even though I'll just tell it in short. So to prove that water is necessary for germination, here they are going to take a 2 beaker A and B. In beaker A they are going to keep some wet cotton wool, they are going to place some seeds, and in beaker B they are going to keep dry cotton and they are going to put dry seeds over there. So after 1 or 2 days, the seed which is present in the beaker A start germinate, and the seed which is present in the beaker B does not germinate because there is no water. So this proves the water is necessary for germination. And the second is to prove that suitable temperature is necessary. So here also they are going to take 2 different beaker A and B. In that they are going to place some green gram seeds on a wet cotton wool on each of the beaker, and one is kept, that is the beaker A is kept under the room temperature and beaker B is kept inside the refrigerator. After a day or 2, what happens? There is a germination seen in the beaker A but not in B. Why? Because the beaker A is kept in the optimum temperature. The necessary requirements are fulfilled by the beaker A. Here beaker B, it is completely kept in the refrigerator, so it does not yield the optimum temperature for the growth. So this proves that suitable temperature is necessary. And the third is to prove oxygen is important. Here also they are going to take 2 beakers A and B, and they are going to place some seeds inside that, and in beaker A they are going to lower a small test tube with water, and beaker B they are going to lower a small test tube with pyrogallic acid which absorbs the oxygen. So after few days, what happens? The seeds present in the beaker A starts to germinate, but the seeds which is present in the beaker containing the pyrogallic acid does not germinate. Why? Because the oxygen present in the beaker was absorbed by that acid. So that oxygen is important for the germination. And the 4th experiment is 3 bean seed experiment. Here I have given you the picture. The picture itself tells you very clearly about the experiment. That is, in a slide, 3 bean seeds are pasted, and one is completely immersed in the water, another one is completely exposed to the air, and the middle seed is off immersed in water and off exposed to the air. So the seed which is in the middle, it's getting the proper water and also the temperature. The seed started to germinate, and the remaining 2 seeds does not, because it does not get the water, one is not getting the oxygen. Okay, the seed which is present in the water may slightly germinate because the oxygen may present in the water, but the oxygen is diffused in the water completely, so the seed cannot completely absorb the oxygen, so that it may slightly germinate, but the complete germination can see in the middle seed. Next, conclusion. So germination. Germination in any plant ends with the formation of seedling. So seedling, it is develops into a new plant. This new plant keep growing and it mature. So once the plant is matured, it's going to produce the flower. This flower is going to produce the seed, and this seed in turns again go for germination, seedling, new plant. So the cycle rotates. So this is what the seed structure and germination. I hope so children, you are all very much clear with this topic. So after this session is over, please go through your book. If you have any doubts, just mark it and keep. And very soon I'll meet you all with a next video with a new chapter, an interesting chapter. Please be safe at home, stay safe. Thank you. Have a nice day.